Good morning, class. Welcome back from your holidays. I want to believe you had a nice time. Enjoy your holidays. Well, in about 111, we enjoyed ourselves. And I believe all of us must have gained one or two things during about 111 lectures. I believe we'll have the same thing too. Enjoy ourselves together in ABIO122, General Biology 2. I'm CKO Idahor. I will be taking animal science component of ABIO122. In that case, we'll look at the animal cells, types of animal cells, and then cellular organelles, cellular components, cellular parts, or cellular inclusions. We'll continue with functions of animal cells, cell cycle, and cell division. And then we'll come back again, meet for revision and exam. So let's look at general structure of animal cells. There are some animals that are actually unicellular, meaning they are just a single cell organism. And such animals are called prokaryotic animals, meaning they have a simple or unicellular cell. A good example is the bacteria. A bacteria on its own is an entity, though it is a single-celled entity. In that case, it has a flagellum or a flagella that will help it in the movement from one position to another. From what you can see here, it has capsule, cell wall, plasma membrane, cytoplasm, ribosome, plasmid, and pili. And then, of course, you have the nucleoid that is equivalent to nucleus in higher organisms. In this nucleoid, you have circular DNA, which is equivalent to the DNA, well-organized DNA in well-organized animal system, that is the multicellular organism. Now, let's compare it with what we have in eukaryotic cells, like in plants, animals, fungi, slime molds, protozoa, and algae. All these are good examples of eukaryotic animals. Now you have the rough endoplasmic reticulum, the nucleus, which is of course not found in prokaryotic. We have it in eukaryotic. In the nucleus, you have the pus, the membrane, and then you have the nucleolus and the nuclear envelope. All these are not found in prokaryotic because this is a well-organized system where the cellular inclusions and the cellular bound organelles, including the nucleus, are found. Now, let's look at the differences or comparison now between prokaryotic animals and eukaryotic animals. Now, the size, the maximum size you can have among the prokaryotes is about 5 micrometers. Whereas in eukaryotes, like in protists, you could have close to 100 micrometer as the least size. Then the type of nucleus you could find in prokaryotes is that of nucleoid, which is not actually a true nucleus. And of course, you have the true nucleus with double membrane in eukaryotes. Then the ribonucleic acid synthesis or protein synthesis takes place in the cytoplasm in prokaryotes. Same in eukaryotes, but then in the cytoplasm, it is at the higher level. For example, if there is a need for protein synthesis, the ribosome will now be used as a messenger, that is mRNA, that will migrate to the nucleus, to where the DNA is, and then copy based on the codon code as directed by the hypothalamus of the brain. And then once the messenger copies it, it is transcribed and translated exactly the way it is. Now, that is done in the ribosome. If, for example, the structure that is required in the system is that of a glucoprotein. Then a glucomolecule will be from elsewhere within the cell, and that of protein synthesized from the DNA in the nucleus that the transferable ribonucleic acid just brought to the ribosome will be assembled in the Golgi body. And now it goes close to the surface of the cell, and then it is picked up through the cell membrane into the bloodstream, where it is taken to where it is required in the system. That is for ribonucleic acid synthesis and protein synthesis. Of course, both will occur in the cytoplasm of the cells. Now, the cytoplasmic structure is quite few in prokaryotes, whereas it is highly structured in eukaryotes. For example, the endomembranes and the cytoskeleton, they are quite highly structured in eukaryotes. Then the movement, cell movement itself. In prokaryotes, the flagella, the flagellum, the cilia or the cilium is used in movement from point A to point B. Well, you may not have this in eukaryotes, that you are, because you are not, the cells are not expected to migrate from a particular position to another position. But then it is possible you have them in case there's going to be a form of interaction between a particular cell and another cell, but there might be no obvious movement of the cells. And of course, you have the mitochondria. There is nothing like mitochondria in the prokaryotes, but you have it, several of it, sincerely, in eukaryotes. You have the chloroplast. It is not also there in prokaryotes, but you could find it in algae and other plants. Then the cell division. The prokaryotes will divide via binary fission. That is, the cell will divide into two daughter cells, forming two bacteria. Whereas in eukaryotes, you have two forms of cell division. That is the mitotic cell division and meiotic cell division. In mitotic cell division, the cells will divide and then still contain the chromosome number, the exact chromosome number of the animal species. For example, goat. Goat has 60 chromosome number. Now, if in mitotic cell division, that is body cell division or somatic cell division, after the division, the cell will still contain 60 chromosomes, meaning in each of the diploid cells, you are going to have a complete chromosome number. But in meiotic cell division, which occurs only in the gametes, that is the gonads, that is the paired ovaries and the paired testes. So the meiotic cell division will now take a phase one, and then the diploid will now undergo another phase of division, resulting in haploid cell formation. That is reduction in the chromosome number, now having each of the cells having 30 chromosome number in case of goat. Meaning the first cells that will result from the cell that started dividing in mitotic phase, now extended through the meiotic phase, will have 4 cells, but at half of the chromosome number. Then you have the chromosomes, which is just a single strand in prokaryotes, but it is double in eukaryotes. The membrane generally in prokaryotes is just the cell membrane, whereas in eukaryotes, it has the membrane-bound organelles and as well as the cell membrane itself. Now, in the form of revision, we have said general structure of animal cell will contain either components, parts, inclusion, or organelles. Whichever you use is correct. And we have two types of cells. That is the prokaryotes, that is associated with unicellular organism, and the eukaryotes that is associated with multicellular organisms. And we said this depends on whichever organization status they have. That the flagellum in the bacterium or bacteria, which is a unicellular organism that could be used in the movements, whereas in eukaryotes, they are more or less in the tissue that will now function together as a tissue, and several tissues aggregating to form an organ playing the same role as an organ. And all organs put together will now form the system in the animal. So when next we meet, we'll continue with cell division. Thank you for your attention.